name is Cavezza, and this is the Gut Microbiome Project. Just for some introduction, the gut microbiome is defined as a proportion of microorganisms in a given environment. In this case, the gut is the environment. Based on the gut-brain connection theory, the gut microbiome affects both mental and physical health. A poor microbiome can influence IBS, colon issues, and various mental illnesses such as depression. The microbiome is most at risk for deteriorating at infancy and old age. It is important to keep a good proportion of microorganisms at these stages. Two studies that we chose to do were IBS and depression. For IBS, Andrew Schreiner researched and showed that the gut microbiome is correlated to diet, environment, and several aspects of health, including obesity, diabetes, and irritable bowel syndrome. His scientists and him discovered that the microbes found in the gut contribute to metabolic functions, they protect against pathogens, and strengthen the immune system. So those with a weakened immune system can get IBS. For depression, Zhang and his team discovered, with the bacterial evidence, that the gut microbiome affects mental health, especially depression. The proportions of actinobacteria and bacteroids alter the amount of serotonin and may influence a person's likeliness to develop depression. Our research question for this experiment was, how does age affect the gut microbiome? Our hypothesis was, as age increases, the gut microbiome becomes more stable and therefore is less at risk for poor gut health. However, when the organism goes into the end stages of life, it then deteriorates. Our rationale is, as infants are low in age, their gut microbiome is only developing, so it is at risk for disease. Meanwhile, when we grow older into the end stages of life, the body also begins to deteriorate, and so will the proportions of the microorganisms, meaning that the middle age is the best for gut health. Our methods and materials. The two animals that we studied were both dogs, and they were 307 and 308. Hoshi was number 307, and she was a dog as well as a flat-coated retriever. She is 6 months old, she weighs 50 pounds, she's female, has not sexually matured, she's fed Mother Hubbard biscuits, table scraps, yogurt, which is dry food and grass, and she's named animal 308. Animal 2 is number 308, and she's Nova, a dog, a flat-coated retriever. She's 4.5 years old, she weighs 50 pounds, is female, and she's fed also Mother Hubbard biscuits, table scraps, yogurt, and grass, and she is Aunt 307. For week 1, we gathered the DNA from fecal samples in order to use the QIAGEN kit and Zymo kit to isolate and concentrate the DNA. The QIAGEN kit is specifically designed for the isolation of DNA from fecal samples. To do this, we added a buffer solution to prevent the DNA from degrading and to bind the DNA to a filter within a small column. We washed away impurities by using a series of washes, wash buffer AW1 and wash buffer AW2. Then we transferred the buffer ATE into the column to instruct the bonding between the column and DNA. To guarantee DNA isolation, we use the following solutions: inhibit EX buffer, buffer AL, lysate, wash buffer AW2, proteinase K, ethanol, wash buffer AW1, and buffer ATE. The Zymo kit is used to concentrate the DNA, 10 times the initial amount of it. The DNA from the QIAGEN kit is bound to the filter in the Zymo column, is rinsed of any impurities, and eluted into a special buffer at 1/10 the volume of the initial. We use the following solutions to guarantee the concentration of DNA increased: chip DNA binding buffer, DNA elution buffer, DNA wash buffer. For week 2, we isolated and concentrated DNA from fecal samples from specific animals. The specific gene we wanted to analyze was the 16S ribosomal gene, which allows us to determine which types of bacteria were present in the organism. It has 9 variable regions and is frequently used to determine classification, such as the genus of an organism. First, we determine the concentration of the DNA, and then we amplify the 16S ribosomal gene using PCR, specifically the V3 and V4 regions of the 16S gene. To determine the concentration of the DNA, we use UV spectrophotometry because DNA absorbs very well at the wavelength of 260 nm. After determining the concentration of DNA at the 260-280 nm ratio, we determine the purity of the DNA. If the concentration was between 1.5 and 2.2, it was red. If it was less than 1.5, it was probably contaminated by proteins, and if it was greater than 2.2, it was contaminated by RNA. We added a series of solutions which added the necessary components for PCR to occur. This includes primers, nucleotides, and DNA polymerase. The primers had to bind to the target sequence of DNA before the DNA polymerase could bind and begin adding nucleotides to the 3' end of the primer. To begin the PCR process, the temperature of DNA must be raised to 95 degrees Celsius. After the primer is bound to the temperature, again is lowered to 72 degrees Celsius. So DNA polymerase could bind and begin synthesizing. To make the PCR reactions, the solutions were put in a thermocycler. Each cycle of PCR potentially doubles the number of the target molecules. In week 4, in week 3, we checked to make sure the PCR reactions from week 2 were successful using gel electrophoresis. Gel electrophoresis relies on the charged particles in the solution to migrate in an electric field. The DNA migrated from the negatively charged electrode, which is the anode, towards the positively charged electrode, which is the cathode. DNA molecules have a negative charge because of the phosphate groups in the backbone, which cause the DNA to migrate towards the anode. We can calculate the size of the DNA fragments by the distance the DNA molecule travels, and it's inversely proportional to the logarithm of its molecular weight. The expected size of the amplicon was to be about 550 base pairs. After the PCR, we had to clean it up by removing the components of the amplification PCR buffer, which include magnesium 2+, DNTPs, and enzymes. Then, any unused forward and reverse primers. The Agincourt AMP pure PCR cleanup system theory is the double-stranded DNA will be bound to solid magnetic beads. The single-stranded DNA with the primers will be washed away with solutions and other impurities. Then we determine the nucleotide sequence of the fecal samples in a large mixture containing all the other student samples. We had to mark a specific sample by adding the last 2 digits of our animal's specific number. We ran an index PCR for 8 cycles since we tried to add a short sequence instead of making a large number of amplicon. In week 4, during the beginning of week 4, all of our previous data was lost and we had to use the past semester's data to finish. The Agincourt AMP Pure XP kit is used to remove the components of the amplification PCR buffers and leave the DNA, which was binding to a magnetic surface, to minimize the amount of DNA that was lost. We used a fluorometric assay to determine the concentration of the amplified sample after the impurities were removed. We did not use UV spectrophotometry because it cannot determine the difference between proteins, RNA, and DNA, and would result in a higher concentration value than what it truly was. In contrast, the fluorometric assay can target a specific nucleic acid and give accurate concentration values. In week 5, the data was received, which contains the results of the gut microbiome's diversity at the following levels: kingdom, order, and genus. Here we have the results for our UV absorbance spectrophotometry. For the older dog, number 308, the concentrations was 29.7 nanograms per microliter, and its 260/280 ratios were 1.67, and for the younger dog, the concentrations were 15.8 nanograms per microliter, and the 260 to 280 ratio was 1.84. The DNA ratio should be approximately 1.8, which indicates the DNA concentration is good. 260 to 280 ratios of 1.5 and 2.2 are considered to be within the acceptable range, and both of the dogs were found to be within this range. Using the concentrations determined from spectrophotometry, we calculated the amount of DNA that will be put into the PCR reactions. Here it shows the number of reads that were given for the samples that we had to use from previous semester. The 07E 307 sample had the total reads of 2,687, and these reads that passed the qualifying filtering were again 2,687, and the percentage of reads that passed through the quality filtering were 100%. And for sample 07E 308, which is the older dog, total reads were 18,028, and all those reads passed with 100%. And for our positive control, there was 2,015 reads, and once again, it was 100% quality filtering. And for our negative control, only 98 reads were done, and it was 100% passed through the quality filtering. Gel electrophoresis here's our picture of gel electrophoresis results. And gel electrophoresis was done to check the products of the PCR reactions. The control shows where the band should be found if the PCR reaction was performed correctly to isolate the 16S ribosomal gene. The negative control showed the DNA present, which indicates there was contamination. The dog DNA didn't show up, so that means there was not enough DNA present to make the band. Moving on to our graphs. At the kingdom level, this graph shows the percentages of classified bacteria shown up in the reads at the kingdom level. For the older dog, 99.02 classified bacteria was found, and in the younger dog, 75.25% was found. However, for unclassified bacteria in the older dog, 0.98% was found, and the younger dog, 24.75% was found. And going more in depth into the order, this graph displays the vast proportions of different bacteria orders. The green bars are for the younger dog, while the blue bars are for the older dog. And at this table here, you can see our percentages. And basically, we found that there was higher percentages for more bacteria at the older dog and lower percentages for the younger dog. And there was a high percentage for the younger dog of unclassified bacteria. And for the genus, this was even more specific to the bacteria. And this graph displays the vast proportions of different bacteria genuses. The green bars, once again, are for the younger dog, while the blue ones are for the older dog. And the same results were found in the genus, that there was a higher percentage is for the different bacteria in the older dog, while the younger dog had lower. And once again, the unclassified percentage was much higher in the older dog. So basically, for our discussion, the DNA extracted from the stool samples was successful. For each sample, we removed 500 microliters of isolated DNA, and after using UV absorbance spectrophotometry, we determined the DNA was pure enough for each sample. For the DNA, the 260/280 ratio should be approximately 1.8, which indicates the DNA concentration. And our ratios were both between 1.5 and 1.2, and so this was good. And the gel electrophoresis was done to check the products of the PCR reaction. The control shows where the band should be found if the PCR reaction was performed correctly to isolate the 16 S-ribosomal DNA. And for ours, it was not, so that would mean that our negative control was contaminated, as well as our dog samples. And during the start of week 4, our data from the original samples was lost, so we were unable to determine, so we had to improvise and use previous samples from previous classes. The positive control had over 1,000 reads, so that passes the quality filter, and 1,000 of those reads passed to the 100% of them passed to the quality filter. At the kingdom level, 99.02% of the older dog was bacteria, while only 0.98% was unclassified. This means that the positive control had little to no contamination, since it was there's expected to be a high percentage of bacteria. So that means our older dog as well has very little contamination. Meanwhile, there's a lot of percentages in our younger dog, which means there might have been some contamination. And the gut microbiomes of the 2 animals we chose for our project had similar kingdom levels, because any animal on the planet can have the same bacterium, and we kind of concluded that as it gets smaller from the order, it becomes more specific and more specific to the genus. So that means that the percentages got more filtered and more different, and as we divided it each up. And finally, our results support our hypothesis that the older dog would have more diversity in the gut microbiome than the younger dog, because we found that at the genus level, the older dog had 6 different genus classifications that were greater than 6%, while the younger dog only had 5 that were greater than 6%, which isn't very good. And at the order level, the older dog had 4 order classifications that were greater than 6%, while the younger dog had only 4, but the percentages were way lower.